All right, guys. I think I've got it working. So a couple of reminders, your test is Thursday, and you're going to be good. Keep up with the reading. Even if you're not taking in every single word, getting the basics out of each section is important. But reading's fair game on the test. All right. The notes are not required, but they're extra cushion points. So the ones for this unit are 40 points. So you do the notes, you get 40 points. You don't do the notes, you don't get 40 points. It just goes in as a excuse, not a zero. So totally up to you, but they are how you get the cushion points. There's not a whole lot of those in here. All right, so let's see. What do you think you know? Yes. Jackson, can you open that cabinet behind you and grab boxes out? Sorry. All right, so here's what's going to happen. You can write with your finger, you can write with the pen. Come up here, and if you think you know something is definitely correct, come fill it in. I only have one pen, though. No, come on. I'm not calling them, just come up here. Yes, if you're sure, come write one in. I don't care. Come on, one person. I don't care. It doesn't matter. I'm not calling on people. You can write with your pen. You can write with your finger. Whatever you want. Now, I'm not saying these are right. I just want to see what you know.
Nice. You failed me.
Oh, sorry, you can write that one.
It's not.
Just go do it. It'll be funny.
You can't read it, but it's there. You can write with your fingers.
I ain't got it.
All right. Anybody else have anything they want to fill in?
Just enough. More than one thing?
Yeah, come on. Use your finger, use your pen. I know, I can't think of it. Yeah, you can all write at the same time. You don't have to wait.
I mean, I have no.
The computer lets you on the video.
I've worn shorts like the past 2 years for my life. Very rapid transition for hands.
I know, it's so hard.
It's phosphate group, not phosphate.
No one's gonna fill in their eyes.
All right, so.
Dude, I don't got it.
There's more stuff. There's more stuff.
Why did you write all this stuff?
Oh, yeah. Somebody else put it.
What do y'all think? Right or wrong?
Questionable? I think everything is right. No, not everything.
Not everything's right, but a lot of it is.
I don't think.
So, we're gonna come back to this after the notes and see what you can fill in or what you can correct. I've got to figure out how to calibrate this. I know there's supposed to be like a. There's supposed to be a fire button somewhere. Oh well. Okay. So are you ready? So if you're ahead in your reading, or you've done any of your reading, you're gonna be like, hey, I remember hearing some of this. We're just gonna hit the high points, review it, make sure you got it. We did talk about this stuff last semester, so a lot of it should be reviewed for you. Are you ready? Everybody have a note sheet? Okay. Can you fill in the first 3 blanks? Protons have what kind of charge? Positive. And neutrons have, how many of you want to say negative because it started with an N? Electrons have a negative charge, and an abbreviation for electrons that I will use throughout the year, E with a negative superscript. Totally okay. Once you, like if you're writing, once you say the word electron and you put E with the negative sign in parentheses, after that you don't have to write the word electron. You can just put E with the negative sign, or you can write it out every time. All right, which of these would you label as electrons? Those, right? Which ones would you label as neutrons? The white ones. And the purple ones are protons. Thank you, Candace. When an atom is missing one or more electrons, it will have what type of charge? So if this atom right now, it has 4 protons, 4 electrons. You need me? Okay. 4 protons, 4 electrons. Positive 4, negative 4, they cancel each other out, so it has no charge, right? If it loses this, it becomes positive. If you lose a negative attitude, you have a positive attitude. So when it loses an electron, it has a positive charge. If it gains an electron, what happens? So when an atom has 1 or more extra electrons, it will have a negative charge. If you hang out with negative people, you become more negative, right? So the more electrons, the more negative. An atom with a charge is called a what? An ion. Now, what charges can an ion have? Positive, or it can be negative. A positive ion is called a cation, and a negative is called an anion. For some people, having a cat is a positive thing, right? So cation, positive, anion. This prefix right here means against, not, so negative. Questions? Feeling okay so far? What do we know about a positive charge and a negative charge? They're attracted to each other, right? So now we're going to take this and we're going to talk about bonding. What electrons in an atom are important for bonding? The valence electrons, the electrons on the outer shell. All the other ones we don't really care about, but the ones on the outer shell are the ones that are going to be important for bonding, and those electrons can interact in different ways. Anybody remember the 3 types of bonds that we can see, or that will form? Yes. What? Okay, ionic bonds, covalent bonds, and hydrogen bonds. So we're going to talk about those real quick. So All right, so let's talk about ionic bonds. How many valence electrons does this atom have? One, right here. And how many valence electrons does this one have? 7. Now, the whole idea is that an atom wants to be stable. To be stable, it has to have a full outer shell. So, for this one to be full, it has to get 8 electrons. And for this one to be full, it has to get 8 electrons. But this inner shell only has to have 2. So, is this shell full? Yes. So, is it easier for this atom to go find 7 electrons or to just get rid of 1 and then have a full shell? If it gets rid of 1, this is now its outer shell and has a full shell. What about this one? It's easier to gain 1. This atom has a higher electronegativity. Meaning it's going to pull electrons toward it. So this electron gets donated here, and it's lost here. So this one gained an electron, so what happens to it? Okay, it has 8 electrons and it gets a negative charge, right? This one lost an electron, so what happens to it? It gets a positive charge. Does it have a full outer shell now? Yeah, because this is its outer shell, it's full with 2 electrons. So now we have a positively charged ion and a negatively charged ion. Are they going to be attracted to each other? Yes, that's your ionic bond. So an ionic bond does what? Okay, so electrons are transferred, right? What would this atom be called? If it's a positive ion, it would be a what? And this one's negative, so it would be a anion. So does anybody know an example of an ionic compound? NaCl, which is our table salt. In fact, all salts are ionic. So ionic bonds it's like being held together with tape. They're held together, but can you pull them apart? Yeah. Kind of strong, but not super strong. Questions on ionic? It's just those positive and negative electrons transferred, now they're going to be attracted to each other. Good girl, bad guy. They're going to be attracted to each other for a little while, but eventually it's not going to last. All right? So next bonds. Covalent bonds. Co means what? Together, both, right? So here we're not going to transfer, they're going to stay together and they're going to share the electrons. They're both going to have the electrons. These are our really strong bonds. These are the ones that we see in all of our macromolecules between the carbons. They're hard to break and they can either be polar or non-polar. What atom only has 1 electron? Do y'all know? Hydrogen. So here we have hydrogen and hydrogen. When they come together, it becomes H2. But what would this look like when they come together? These electrons are going to be shared. Well, they only have 1 electron. How many do they want to have a full shell? 2. Well, now they're sharing both of these. So does this one have 2 in its outer shell now? And this one has 2 as well because they're sharing them. So that's covalent. Non-polar, they're equally sharing those electrons, and I know it says that in there, but I need to make sure you understand. If we say it's covalent, usually we're referring to a non-polar covalent bond, and they're equally sharing them. But sometimes we have polar covalent bonds. And polar means that we're going to have two different ends to the atom, like the Earth has a North Pole and a South Pole, right? So we're going to create, or two different ends to the molecule. We're going to create two ends with different charges. But polar means that we're not going to share them equally. One atom is greedy. What's the word? What's the science word for saying an atom is greedy? It's going to try and take electrons. Now, an atom that's electronegative, there are a bunch of them. And as we go through the semester, I'm going to say, this is a biology rule. When you get to chemistry, Mr. Gaffer is going to say, that's not true. I'm not lying to you. I am teaching you within the scope of biology. And when you get to chemistry, you start dealing with other elements that don't really affect in biological systems, okay? So biologically, the most electronegative atom is oxygen. There are others that are more electronegative, but biologically speaking, oxygen is the most electronegative atom. Okay? That means it's the most what atom? Greedy, which means it's going to try and take what? Electrons. So here's oxygen. How many electrons does it have in its outer shell? 6. How many does it need? It needs 8. So it needs to get 2 more. How many does this hydrogen need? How many does this one need? 1. So if each of these share their electrons with this one, would they all have a full outer shell? So here's what happens. Here's our oxygen. These are our hydrogens. See these electrons right here? See how it's kind of misshapen? Oxygen is pulling these electrons toward it. So these electrons are closer to the oxygen and further away from the center of the hydrogen. And so it creates this kind of bent shape. And it creates what we call a polar molecule. And the most common polar molecule we're going to talk about in biology is water. But this oxygen atom is trying to steal the electrons. Hydrogens are holding on for dear life, but these electrons are closer to the oxygen. Electrons have what kind of charge, Ethan?
Negative.
Negative. So if these electrons are closer to the oxygen, this oxygen is going to have a what? It has what we call a partial negative charge. That little symbol, it means partial. All right, it's slightly negative, and these hydrogens are almost losing their electrons, so they almost have a what charge? Positive. So they have this partial. How can I draw that? I can't draw. Partial positive charge. So now we have this water molecule that has a positive end and a negative end. Does it have poles? Like a North Pole and a South Pole, but it's got a positive pole and a negative pole. So 13, answer it. And answer 14. What do you think? So which atom will have electrons most of the time? The oxygen. And if electrons spend more time at one end of the molecule than the other, what does it do? Makes a partial negative end and a partial positive end. Or poles. What is an example of a non-polar covalent bond?
Not like that. Yeah, that's hydrogen.
Okay, between two hydrogens, between two carbons, between a carbon and a hydrogen. Anything with carbon is going to be non-polar covalent. So let's look at hydrogen bonds now. Now, hydrogen bonds are the ones where you can stick your finger in the water and what are you breaking? Hydrogen bonds. They're the ones that create the surface tension. So hydrogen bonds are very weak, but they're also very important. So obviously I can't go back to number 12, but you can. So if you draw a second water molecule, draw a hydrogen bond there. Or you can draw two water molecules wherever you want on your paper and draw a hydrogen bond between them. Draw two water molecules, or use one from number 12 and draw a hydrogen bond between them. What do we know about opposites? So can the hydrogen bond be between this hydrogen and this hydrogen? Why? They're both positive. So the hydrogen bond has to be between a what and a what? An oxygen and a hydrogen. Notice I'm drawing a dotted line, not a solid line, because it is weak. What type of bonds within a molecule can lead to hydrogen bonds? Hydrogen bonds are bonds that occur between molecules. So hydrogen bonds occur between molecules. Where do covalent bonds occur? Within a molecule, right? Within atoms in the same molecule. Questions on hydrogen bonds? Where else can we find hydrogen bonds? Who said it? Yeah. Where in the DNA? Do you remember? Right. It's holding together the letters between the bases. So we see hydrogen bonds between water. We see them in DNA. They're also present in proteins, but DNA and water are the most common examples used there. Questions on bonds? I feel like this was pretty much review of last semester. Y'all agree? Don't remember it? Way back in August? Do we need to do some jumping jacks?
Yes.
Oh, the books. This is why I don't lecture anymore. All right. Molecules that contain carbon. What's the word we use for them? So all organic molecules contain carbon. What else do they have to have? Hydrogen. And life is based on what molecule? We don't have, we can't have life. Carbon. So, let's do this real quick. How do we get the carbon we need for all living things? Not say respiration. Photosynthesis. Because what happens in photosynthesis? Plants take in what? They take in light. They take in carbon dioxide from the atmosphere, and they make carbohydrates, which then can be broken down and converted into, yes, ATP. But the carbon we can use to make glucose. We can use it to make lipids. We can use it to make proteins. If plants don't take carbon dioxide out of the air, can we get carbon? No. Because how do we get our carbon? We have to do what? Eat. But if the carbon dioxide is never taken out of the air, it's never in the food. We don't have any food. We can't get any carbon dioxide. So photosynthesis is essential to life, not just for the energy, but also for the carbon. So some things you need to know about carbon, why it is so important. It has 4 valence electrons. Which allows it to form 4 stable covalent bonds. With carbon atoms or atoms of other elements. So if you see, here's carbon. It's sharing electron. Oh my goodness gracious. I can't draw dots apparently. But it's sharing 2 electrons with each of these. Each of them being a nonpolar covalent molecule. The fact that it can form, or nonpolar covalent bond, the fact that it can form 4 covalent bonds makes it very versatile. The fact that it can form covalent bonds with lots of different molecules allows for versatility, allows for a lot of different formations, a lot of different structures, and a lot of different types of molecules to form. What elements does carbon commonly bond with? Okay, oxygen, hydrogen, nitrogen, phosphorus. Can carbon bond with carbon? Does this? So, but we're going to add an S to it. It will also attach to sulfur, which we do see in living organisms as well. Below that, in the box, write what each of those letters stands for. Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, right? All right, so a couple of things I need you to understand. When we draw molecules with carbon, we can draw them like this. How many carbons are in that molecule? 4. How many covalent bonds? 3. Now I can also draw this same molecule like this. Four points. There's a carbon at every end and every bend. So there's a carbon here, 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 and here. Does that make sense? I'm telling you because you're going to see structures drawn both ways. Carbons can form double bonds. If they're double bonds, you'll see 2 lines there. That means it's sharing 2 pairs of electrons. On this, if I wanted to show a double bond, well, I would draw 2 lines there. So this would be called a chain. So carbon can form chains. But sometimes carbon, because it can form 4 bonds, it can form rings. But I can draw this like this as well. Every end, every bend, there's carbon. If I put an O here, it means that bend, is there a carbon? No, there's an oxygen. Sometimes from those rings or even the chains, we can have branches where another carbon comes off of. Is this an end? So there's a carbon there. So carbon-based molecules can form chains, rings, and branches. So you could see molecules drawn like this. You could see them drawn like this. You could see them drawn shorthand with those lines.
So when you see like in that house shape, that's only for carbon, like, or like the bent lines, that's only for carbon.
Yes, if it's a different element. So if we had this, and I could, if there's an oxygen there, I'd have to write that. I couldn't assume that there was an oxygen.
Okay.
But if there's just a bend or an end, it's a carbon. Questions? Still okay? So now we're going to take all these atoms and we're going to put them together and talk about all these molecules that form. And they get properties based on the elements that are present and how those elements are arranged. And if you did your reading, you saw functional groups. That might be a nice reading. Chapter 4. It's a nice reading. Functional groups. Are you ready? Are a collection of atoms that, when attached to carbon backbones, and by backbones I just mean a chain of carbons, a ring of carbons, give the molecule specific properties. So when we see certain atoms together in a molecule, we can go, oh, this part of the atom, this part of the molecule is polar, or this makes this molecule acidic. There are a lot of functional groups. And when you open your book, it takes up an entire page. We just have to memorize all of them. You have to memorize 4. And they're right here for you. So the first one is called carboxyl. And it is a carbon double bonded to an O and single bonded to an OH. When it says R, this just means the carbon atom that it's attached to, or the carbon molecule. That says rest of molecule. So R is not really part of it. So the carboxyl is really a COOH. Carbon double bonded to an oxygen and attached to a hydroxyl, or an OH. So one of them you have to remember is carboxyl. If you notice, it's the only one that has carbon as part of its functional group. The next one is an amino group. Guess what it might be a part of? Amino acids. The next one is a phosphate group. Where do we see phosphate groups? What molecules? DNA? On the nucleotides? What else? Okay, DNA and RNA, absolutely. On the nucleotides, where else? What type of lipids? Phospholipids. We also find them in something that y'all all need a little bit of right now. ATP, right? See, y'all got there? You got sugar, energy, ATP, you got there? Phosphate groups are PO4 or they can be PO3. If they're PO3, they usually have a negative charge to them. And the last one, sulfhydryl. And you can't see it on mine, but it's an SH. So when these groups of atoms attach to a molecule, it gives it certain properties. When carboxyls attach, it can allow acids to form. Aminos, the amino group, are considered basic by themselves and they can allow bases to form. Both of these are part of an amino acid. So the amino name comes from this, the acid name comes from the fact that it has a carboxyl group. Phosphate can make it acidic, it can also make it charged, meaning it can give a molecule a charge. We give it a negative charge, which would make it attracted to what? Positive. What part of the phospholipid is attracted to water? This is where the phosphate group is. So it has a negative charge, but that makes that part of it attracted to water. Yes. And then we have sulfhydryl. You're going to see sulfhydryls in proteins on amino acids, and they can give a polar quality or a property. So this, guys, is memorization. I'm not going to give you this chart and ask you to fill it in, but you do need to memorize and you do need to be able to recognize them. All right. How many of you heard the words polymer and monomer before? All of you should be raising your hand from last semester. So talk to your partner, write in the definition for polymer, write in a definition for monomer.
Monomer is a building block of a polymer.
The polymer gives us strength.
You said monomer? Monomer is a building block. Monomer is a building block.
monomers put together?
Poly means many. All right, and Ethan, give me your definition for monomer.
We just like put that, they're not connected.
Okay. What else? They're not connected?
Here's the building blocks for monomers.
Y'all agree with those? All right, so now take what you learned last semester and the pictures and write a definition for dehydration synthesis or condensation reaction and for hydrolysis.
They're not defined yet.
Water loss due to
Water's lost.
All right, so dehydration synthesis. What is happening in dehydration synthesis?
water is being lost to create one.
Okay, so water is being lost.
I said water is formed by taking hydrogen and oxygen out of the molecule.
Okay, so water is being formed, and what else is being formed? Gavin, what'd you say?
Bonds.
A bond. The purpose of dehydration synthesis is not the water. The purpose is for two monomers are connected by a covalent bond by removing a water molecule. If yours says something like that, you don't have to have that exact wording. So if you look, here's a monomer, here's a monomer. I'm going to take a hydrogen from this one. And if I take a hydrogen, does that mean this oxygen next to it needs an electron? Yeah, because it was sharing one with hydrogen. So when I take that away, that oxygen now needs it to share an electron. And if I take this away, now there's a carbon here that needs to share an electron. So what happens? This oxygen connects to this carbon, form a covalent bond, but I'm taking out H and OH, which gives me water. The reason it's called dehydration synthesis or condensation is because we get that water molecule. What is condensation? When you go outside with your cup of sweet tea, all the water from the atmosphere forms on the outside of it, right? We see drops of water form. Or we're dehydrating it, we're removing water. Now we also say this is an anabolic reaction. Means we're building something. Anabolic reaction. Putting something together. Synthesis tells us that we're building something, right? Now we have hydrolysis. Hydro means what? Water. Lysis means break down, it's a split. So in this one, polymers are split into what? Into monomers by adding a water molecule. So here's what happens. We have this oxygen here. I'm going to add this water molecule. It's going to cause this bond to become unstable. The water molecule will split and we'll add the hydrogen to one side and the hydroxyl to the other, and those monomers are now broken apart. It takes apart that covalent bond. Guess what else it takes a lot of to break that? ATP, energy. You have to have a bunch of energy to break it down as well. So this is a catabolic reaction. What do you think it means? Breaking down. Now, I know this sounds maybe silly, but I was in college chemistry and we're learning about catabolic anabolic reactions. That was the unit we were on as the same year that the World Trade Center got hit. And I can just see the image of those towers falling apart into all its little pieces. It was catastrophic, right? So a catastrophe, something's falling apart. Catabolic, it's falling apart, it's breaking down. So I try to give you little ways to remember it. If it doesn't work, just ignore it and say it's more weird and move on. Doesn't hurt my feelings. All right. So this little dude right here is our monomer. Here's our polymer. Would you agree that this could be a monomer and this could be a polymer? I want to know how many water molecules were formed to create this. Don't say it out loud, just see if you can figure it out. If you think you know the answer, raise your hand. Victoria, what do you think? What? 9, absolutely. How many of you thought it was 9? Or you thought, well, there's 10, maybe there's 10 of them. Because we're counting the number of bonds that form, right? So for each one of these bonds to form, we would have had to get a drop of water. So could I have had 1,999 molecules up here? Or monomers? How many droplets of water would I need to make a polymer made of 1,999 monomers? 1 less. It doesn't matter what number they give you, it's always going to be 1 less. What process formed these? Dehydration synthesis. What's another name for that reaction? Condensation reaction. If I wanted to break this down, what reaction would have to occur? How many molecules of water would be needed to break this? 9. Another term for hydrolysis is digestion. What are we doing when we digest our food? Are we breaking it down into smaller pieces? Absolutely. All right. 4 categories of organic molecules. What are the 4 categories? What else? Proteins, lipids, and nucleic acids. What elements are in carbohydrates? What elements are in proteins? They're not in order. Carbon, hydrogen, oxygen, and nitrogen. What about lipids? And nucleic acids? Remember this? Cho-cho, chomp, right? So we're going to go through each of these pretty quick. So carbohydrates, elements present. Cho. Carbon, hydrogen, oxygen. Does anybody remember the ratio? 1 to 2 to 1. So what's an example formula that we should all have memorized for carbohydrates? C6H12O6. Does that fit the 1 to 2 to 1 ratio? Absolutely. That is the formula for glucose. It's also the formula for fructose and for galactose. They all have C6H12O6. It just has to do with the way they're arranged. So what type of bonds form between one monomer and another monomer? Covalent bonds. And for each of these molecules, you're going to learn a special name for that covalent bond. It's still covalent, but we give it a special name because we like to make your life difficult. So it's called a glycosidic linkage. Glyco means sugar, so it's a linkage between sugars. So monomers of carbohydrates, what do we call them? Monosaccharides. That's a C. Mono means one. What does saccharide mean? Sugar. So monosaccharide is one sugar. Another way these are referred to are your simple sugars. What's the function of monosaccharides? Okay, they do build polymers, but we use glucose. It's for quick energy. It's very easily processed for energy. This is why when little kids eat lots and lots of candy, they get lots and lots of simple sugars, which they can then use for energy, and they get very what? Hyper. This is why parents don't like to give their kids high fructose corn syrup. Now, 3 examples of monosaccharides. There's glucose, there's fructose, and galactose. Fructose. Okay, high fructose corn syrup. Corn is what type of organism? It's a plant. So fructose is made in what? Plants. It kind of sounds like fruit, right? Glucose is made in all living organisms. Galactose, what do you think it's part of? Animals, because it is used in milk. Now, there's 2 types of glucose. There's an alpha and a beta. What's the difference between the alpha and the beta? Good. So in the alpha, the hydroxyl is here, and in the beta, the hydroxyl is here. Hydroxyl goes up in the beta, and it goes down in the alpha. Y'all might think, okay, that's silly, but you're going to see why it's important in just a second. Couple of fun little facts for you. You probably don't think it's fun. But when you take sugar and you put it in water, what happens to it? It dissolves. Why does it dissolve? It's because it loses this ring shape and it becomes linear, which makes it more easily attracted to the charged ends of the water molecule. Carbohydrates are polar, or at least have some polar properties. Now, if you see this shape, you should think carbohydrate, okay? If you see os, you should think what? Carbohydrate, sugar. So now we have what are called dimers, which means, di means what? 2, so what do we call dimers of carbohydrates? Disaccharides. Examples of disaccharides, we have sucrose, which is made up of a glucose and a fructose. Sucrose is our table sugar. That's the white stuff you use when you bake. Lactose is glucose and galactose. Where do we find lactose? In milk. And people who are lactose intolerant can't break that bond right there. The bond between those two molecules, that's what they can't break. Maltose is glucose plus glucose. This is a form of sweetener that we use. We do see it naturally occurring as well, but if you've ever had a malted milk ball or a chocolate malt, they're using maltose instead of sucrose as sweetener. It gives it a different flavor. What process formed these? Formed by dehydration synthesis. All right, then polymers are called what? Polysaccharides. Examples, we have starch. Another name for starch is amylose. Notice that OSE, it tells us it's a carbohydrate. What organisms can make starch? Plants. Think white part of potato, right? Grains, pasta. Its function is energy storage. In plants. Then we have glycogen. And this is energy storage in animals. And if you notice, starch is just a single line. Here we see glycogen, and it starts to have some branches coming off of it. Third one, we have cellulose. Its function is for structure. Where do we see cellulose? In plant cell walls. Now, cellulose is also called dietary fiber or insoluble fiber. Dietary fiber or insoluble fiber. We can't digest fiber. People take fiber supplements to help keep them regular because they can't digest it, so it passes straight through their digestive tract and it pulls other things with it. But we can't digest it. Then our last one, we have chitin. Chitin is also for structure. I can't get down there and y'all see it. So it's structure. Where do we find chitin? What? Okay, yes. So exoskeletons of arthropods. And arthropods are insects, shrimp, crawfish, are crustaceans. All of those have chitin in them. And it's also in fungi cell walls. We can't digest this either. So all of that would fall into the category of insoluble fiber. Mostly cellulose because while we do eat mushrooms, we can't digest the cell walls of mushrooms, but how many of you have ever eaten a crawfish shell or a shrimp shell? Yeah, it's not good. And you could find it again. if you looked. Don't do that though. So see if you can answer these questions. Talk with your partner.
The present of food oxygen oxygen hydrogen. All these are the same, whatever. Until you look at what each on the outside looking
Discuss it with the people around you, please. You need to learn to talk to them. You can't talk, you never get a new seat.
You're picking on one because
On the, like, some places have like the like some of them are upside down, like right here. Or the one of them is upside down.
I do know. I do know. My bad.
They can't.
You did say oh,
oh wait, look.
The beta glucose and the alpha glucose is called the boxes. So that's the amylase, or the amylase has like all alpha glucose. Cellulose and then alpha. But I like it.
See how these oxygens are
You're saying
flipping
the amount that's all.
opposite, just because that's how the betas have to match each other. All right, guys, so let's see what you got. So alpha and beta, what did you write down? You wrote down about the location of the hydroxyls, right? They're opposite. Then you get to starch and cellulose. What's the difference between starch and cellulose? So starch is made of alpha glucose and cellulose is made of beta glucose. So now it says that humans can digest starch, but we can't digest cellulose. Why? What do we need to help us break down these molecules? Enzymes. Guess what we don't have? An enzyme to break the bond between two beta glucose. So for humans, just remember which one we can break and which ones we can't. Beta is bad. It's not really bad, but we can't break it down. All right. And what reaction is used to form disaccharides and polysaccharides? Dehydration, synthesis, or the condensation reaction? All right, let's talk lipids. What elements are present in lipids? Carbon, hydrogen, oxygen. And ratio. This is Miss Moore's ratio. Has no numbers involved. A lot, a little. If you want the mathematical one, 1 to 2 to less than 1. But a lot, a little. If you're looking at a molecule, and all you see is carbon, hydrogen, and oxygen. You just see CHO. And you see carbons and hydrogens, but you see just a little bit of oxygen. Can it be a carbohydrate? No. So remember how I told you there's going to be covalent bonds with special names? What was the special name for the covalent bonds between monomers and carbohydrates? Glycosidic linkage. So now in lipids, it's called an ester linkage or an ester bond. Still a covalent bond. We just give it a special name. Now, lipids are different in the fact that we don't, it's not really a polymer where it's a chain of a bunch of the same molecules. So some scientists will classify it as a polymer. Some people will say lipids aren't really polymers because it's not repeating units. But monomers, or the subunits for lipids, we can have fatty acids, and we can have a glycerol. Let's talk glycerol first. Glycerol is 3 carbons, each with a hydroxyl attached to it. So 3 carbons, each carbon has a hydroxyl attached to it. That is called a glycerol. Now the fatty acids can be one of 2 things. Saturated or unsaturated. Which of these would you call saturated? Okay, why? Okay, they are straight. But what does saturated mean? Okay, when we say we're, like, you jump in the lake with all your clothes on, shoes and everything, you get out and you're like, my clothes are saturated.
You're soaked.
You're soaked. You're full of water. Can you hold any more? No. So a saturated fatty acid is full of hydrogens. Is there any place to add another hydrogen? No. It is straight. It's also solid at room temperature. Tend to be your animal fats. But look at this one. Is there a place where you can add more hydrogens? Are there empty spots?
Yes.
Right here. So there's hydrogens missing, so there's a double bond right here. Everywhere there's a double bond, we see a bend. And this is called unsaturated because there are places we can add hydrogens. And they are liquid at room temperature. These tend to be our plant fats. Think about what we get from plants. What do we use to cook with from plants? Vegetable oil, is it liquid or solid? Liquid, right? What fat do we use to help cook with from animals? Okay, bacon grease. At room temperature, it's solid. What else do we use from animals? Butter, right? Butter, is it solid at room temperature? It doesn't become liquid till we heat it up. It would get soft, but it doesn't become liquid till we heat it up, right? What did you say? Yeah, like the white part on bacon or that fatty part on steak. You can let it sit there all day long at room temperature and it's going to stay solid. I was I'm making icing for a cake. And I have to figure out the right ratio of butter to sugar to Crisco and all this stuff to make the icing to where it won't melt off the cake, right? And Crisco is plant. It's shortening, but it's from a plant. So as it gets a little bit warmer, guess what happens to it? Starts to kind of liquefy a little bit. So I have to be careful what I use. Instead of using Crisco shortening, I could use animal fat shortening. And guess what's going to happen? My icing is going to stay solid even as it gets warmer, like in Louisiana. So I was actually using this today, or yesterday. All right, how else could I draw that saturated fatty acid? But is it a ring? Could I do this? And all of these ends and bends would represent the what? Carbons. Wherever there's a carbon, if I don't put anything else, you're going to assume there's hydrogens attached to it and it's stable. But I could draw it like that. How would I draw this one? Have a double bond there, right? Right here I have a COOH. What is that? Carboxyl.
Yeah.
So they have a carboxyl group. All right. What is this part right here? What am I boxing on my polymers? 3 carbons. The glycerol. And what are these? Fatty acids. How many fatty acids? 3. This is a triglyceride. And its function? Long-term energy storage. Fats store a lot more energy per gram than carbohydrates because of all the carbon-hydrogen bonds. Carbon-hydrogen bonds are high-energy bonds. See a lot of carbon-hydrogen bonds in that picture? Yes. A lot more than we would see in a carbohydrate. What is this one? Phospholipid. And it has a phosphate head. That is polar. And what does it have down here? What are these two things going to be? Fatty acids. And they are nonpolar because all lipids are nonpolar. All lipids are nonpolar. Write it down, give yourself a note somewhere, but make sure you know. What can you tell me about the head? What's the word for that? Hydrophilic. And the tails? They're nonpolar. They are hydrophobic. Does anybody remember the word when something is both hydrophilic and hydrophobic? Nope. Amphipathic. Phospholipids, what are their function? They make up the membrane bilayer, right? Cell membrane, nuclear membrane, organelle membranes, but they make up that bilayer. And the last one, cholesterol, or steroids. You see the honeycomb type shape? If you see this honeycomb type shape where there's 4 hexagons and pentagons put together, you need to think cholesterol or steroid, functions, hormones. It's also in the cell membrane. It's a hormone, it means it's a chemical messenger, right? All right. Draw a phospholipid bilayer. Do this bottom part first. On the bottom, you're drawing a phospholipid bilayer. Bilayer tells us what? 2 layers. If I put water here and water here, the heads would be attracted to the water. There's bilayer, right? Agreed? Now, if I just were to drop a bunch of phospholipids in a beaker of water, we could see them arrange themselves like this. Where's the water? Is there any water on the inside? Why do they arrange themselves like this when we form a cell membrane? Because there's water on the inside. This is called a mycel. Anybody ever use micellar water to wash your face? To clean your face? The reason it works is because it has phospholipids in it. And it's mixed with water, and when you put it on your face, guess what it'll attract to the middle? All the oils, the fats, all of that will be pulled on the inside of it and pulled away from your face. So that's how micellar water works. All right, let's see if we can fill these in real quick. Bonds between glycerol and fatty acids are called what? Ester bonds or ester linkages. Polymers are formed through the process of what? Or condensation reaction. Cholesterol is used in the body to make hormones such as what? Insulin's actually a protein, but it is a hormone. Estrogen, testosterone, or sex hormones. What do hormones do? They send signals or carry chemical messages through the body. So, and hydrogen-carbon bonds are high-energy bonds. Why are lipids more efficient energy storage than carbs? So lipids have more carbon-hydrogen bonds than carbs. In fact, if you look at a fatty acid, where all these are carbons, right? And what are attached to the carbons? Hydrogens. It's called a hydrocarbon chain. All right, homework tonight. is to read chapter 4, these pages, chapter 5, or chapter 4, 57 through 59, 63, 64, chapter 5, 68 through 75. It should cover the stuff we talked about, so it should be a little bit easier for you to understand and read and go through. Work on your Cornell notes. Guys, we will wrap these up tomorrow. They won't take a long time. Y'all go ahead and pack up so that I can come clean your desk.